Very good. Well, good morning, everyone. Nice to see you this morning. We're continuing on this excursion of oxidative stress. Remember that we saw yesterday, unfortunately it wasn't recorded, but this phenomenon involves an overproduction of reactive oxygen species, and that's a broad umbrella term that includes free radicals and also various non-radical species. Now, for the next few lectures, I want to look at why oxidative stress is of interest to someone like myself, a chemical toxicologist, interested in the chemical damage that toxic substances do within tissues. And in this particular lecture, we're going to focus on DNA as a target for free radicals. And then we're going to look at protein damage on Thursday and then lipid damage on, I think, Monday next week. Now, DNA damage is particularly important and I think particularly interesting, and in terms of being a target for oxidative damage. And this is something that's often ignored in molecular biology courses and biochemistry courses, where they, focus on all the various fascinating functions of DNA. But what they don't talk about often is the fact that DNA is susceptible to damage by toxic substances, and that's where toxicologists have a particular interest. And there's some hints that this sort of damage is actually quite important biologically. And Denham Harmon proposed the so-called free radical theory of aging a long time ago, back in the 50s, but we now think that's probably a bit of an oversimplification, but certainly in the sense that free radicals are not the central driving force in aging. But nevertheless, it is true that as we get older, we do accumulate a lot of oxidative damage in various components of our tissues. And DNA is no exception to that. So even though it's sequestered in the nucleus, and you might think it's sort of like the Fort Knox, protected from invaders, in actual fact it's not protected. And we'll look at some of the factors that make DNA vulnerable to free radical damage. But there are a number of hints that this sort of damage is significant in terms of a potential driver of cancer risk. And just in the fact that cancer tends to be an age-related condition, it tends to plague mammalian species in the latter portion of their lifespan. And this has led people to think that perhaps there is this role for accumulation of genetic damage by free radical species as a function of oxidative metabolism and respiration in tissues. Remember how we saw yesterday that those processes of respiration aren't fully efficient, that the consumption of oxygen, the reduction of oxygen by the respiratory chain in the mitochondria isn't 100% efficient, and it releases these damaging species. So we'll look at evidence and explanations for why DNA damage might be a consequence of oxidative stress. Now let's just identify then some basic features of the sort of damage that can occur during exposure of nucleic acids, and particularly DNA, to free radicals. And one of the things that makes DNA vulnerable to free radicals is the fact that it has this strong negative charge on the sugar phosphate backbone. It's just a feature of nucleic acids. Makes it very hard to get nucleic acids into cells because they have this permanent charge on the sugar phosphate backbone. It's one of the things that makes mRNA therapeutics pretty difficult to get mRNA into cells because it has a strong negative charge on the sugar phosphate backbone. And from a toxicology point of view, this means that there's an affinity for positively charged cations, so things like iron and copper that we talked about yesterday has been catalysts of Fenton chemistry, degradation of hydrogen peroxide. These are stuck on the sugar phosphate backbone of DNA, and this means that any hydrogen peroxide that makes its way into the nucleus, and being a very small molecule, peroxide can quickly diffuse through cell membranes, and it can reach these chelated metal ions on the sugar phosphate backbone of DNA and undergo that Fenton chemistry. So this generates hydroxyl radicals right in the heart of the DNA molecule, and remember, it only exists for about a nanosecond, but that's long enough for it to attack the DNA molecule. Okay, so this is a very conspicuous characteristic of free radical related DNA damage, this oxidative modification of both bases but also the sugar phosphates, backbone itself and the sugars. Okay, so we can summarise the types of damaging events that occur when you cook up DNA with a free radical generator. We're actually going to do this in our lab in a few weeks' time. We'll be screening our compound libraries for their ability to protect against strand breaking. And strand breaks occur when you get cleavage of the sugar phosphate backbone. So you see the sugar groups here, the deoxyribose attached through the phosphodiester linkage. These can be damaged and cleaved, causing a strand break, and if they occur on two adjacent strands in close proximity to each other, then you get a double strand break, and they're particularly nasty to cells because they're hard for them to repair. But that's only some of the damage they can occur. You can also get damage to the sugar itself, sugar oxidation. You can get, and then that doesn't have too many consequences for cells in terms of information storage capacity of DNA, but at the top, those top two, oxidative damage to bases and the loss of bases, that really does have consequences for the ability of DNA to store information, genetic information stable. So that's the whole purpose of DNA. It's a digital system really for storing biological information, and it needs to be do that well and effectively with high accuracy, high fidelity, and so any damaging event that damages the structure of the core informational components of DNA, the bases, is really going to have potential adverse consequences in terms of things like cancer and other conditions. Okay, so one of the characteristic outcomes then of cooking up DNA with a free radical generator is the loss of a base. And this is really quite common. And here we've got a simple diagram showing a GC base pair, one of the two essential base pairs that Watson and Crick discovered hold the double helix structure of DNA together. These strong hydrogen bonding interactions between these two bases, the guanine, the purine base, and cytosine, a pyrimidine base. When it's exposed to hydroxyl radicals, we can get oxidative damage to the guanine in particular, also to the cytosine, but in this case we're showing the damage to the guanine. And what this often does is it weakens, or the term is labelizes, it just weakens the strength of the base, the chemical bond attaching the base to the deoxyribose sugar. So it weakens it, and the base can undergo spontaneous release. So this generates what we call an apurinic site, or an abasic site. Just means that A means no, basic, no base. There's no longer a base there, and we've got this, the sugar phosphate backbone is still intact, but the base is missing. So there's no informational content at that point. So this is actually not that rare, and the guanine base is particularly vulnerable just to spontaneous cleavage of the N-glycosyl bond linking it to the sugar phosphate backbone. So abasic sites do arise spontaneously in the DNA, and this presents problems to the cell because when DNA is being replicated by a DNA polymerase, it comes along, screaming along the DNA, processing the template very rapidly, and it comes to a basic site. It's not giving any information to the polymerase about which base to put in there to keep making the DNA molecule. So what it typically does in mammalian species, it follows what we call the A rule, and just to keep DNA replication going, the DNA polymerases often just stick an adenine in an empty space just to allow replication to proceed. So this can corrupt the informational content of DNA, as you imagine, because adenine, it's only one of 4 bases, so you're actually downgrading the information content at that particular site in the genome. Okay, so this results in what we call an N to A substitution mutation, because adenine is another purine, so we're substituting one purine guanine for another adenine, and so this is a substitution, and N stands for a nucleotide being replaced with an adenine. So it doesn't have, depurination doesn't just occur on guanines, that's why we use an N here, but it's basically whatever nucleotide is being depurinated is being replaced with an adenine opposite the adduct. Okay, so what can happen then, as I just indicated, was during normal replication, a guanine is paired with a cytosine, a normal Watson-Crick configuration. But in the case of an abasic site where the base is missing, many polymerases, not all of them, but many polymerases will come along and just stick an adenine in there and keep going. So this results in a position of A on the insertion of A on the opposite strand of the molecule, and so in that opposite strand would call that a C to A transversion. And then in the next round of DNA replication, that A will be paired with a T in that strand, so you can also report the mutational event in that strand as well as a substitution to T. Okay, so how do we know all this stuff? Well, a lot of the information concerning the types of mutational events that happen in oxidative stress when you damage DNA goes back to some work that was done by radiation chemists and biologists in the wake of the World War II, so that rather sad event when those nuclear bombs were dropped on a couple of cities in Japan. That was associated with increased risk of tumours in survivors, and it wasn't just the Japanese victims, but also service people from the military, and also in Australia too. We did a lot of nuclear bomb testing out in the desert, and people that were exposed to that as workers came down with higher rates of tumours. And so there was a lot of interest in what is ionising radiation doing to DNA, and so radiation chemists began cooking up DNA solutions with ionising radiation, things like X-rays and gamma rays, and just isolating the products. And they've identified over 35 different bases in DNA that occur as a result of irradiation. And as I mentioned yesterday, most of the damage in cells that's inflicted by ionising radiation isn't done by the radiation, but by hydroxyl radicals produced by radiolysis of water. And so a lot of the chemistry that was figured out in radiation-treated DNA subsequently was found to be relevant to just normal organisms living in a non-irradiated environment like ourselves. And so the same chemistry that was found to be associated with exposure to ionising radiation also occurs at a background level in you and I, just as a consequence of respiration and the formation of reactive oxygen. Okay, so this is what made oxidative stress research, I think, get a bit of a shot in the arm when a lot of this chemistry that was initially worked out by radiation chemists, when people developed sensitive assays for measuring some of these DNA damaged products, they found them, lo and behold, in controls, people that hadn't been exposed to radiation. That's what made people interested in oxidative stress, the importance of it. Now, because there's so many bases formed, oxidised bases formed, so we go from 4 nitrogenous bases, and remember the 4 nitrogenous bases, as I've already indicated, are the two pyrimidines, cytosine and thymidine. So these are six-membered rings. And then there are the two bicyclic structures, the purines, adenine and guanine. Okay, so the diversity of chemical damage to these four bases results in over 35 products being detected in and formed in radiolytically damaged DNA. So this means, as toxicologists, we want to understand the individual properties of those various products of oxidative damage. And to do that, it took a long time before we had this capability, but it really took the advent of molecular biology and the ability to modify genomes and cut DNA and rejoin DNA with ligases and things. It allowed us to start looking at the DNA damage, the mutagenic properties, the ability of each of these 35 or so DNA damaged products. How mutagenic is each one of them, and are some of them more important to driving corruption of the sequence of DNA than others? Okay, so this is really cool research capabilities, and the idea is you want to make not only do you need good molecular biology skills, but you also need very good organic synthesis skills, because what people needed to do was make a short stretch of DNA that contains a damaged base at a very defined site in that piece of DNA, and then you go and clone that piece of synthetic DNA into a genome and see what happens when you replicate that genome in a biological system. So this is usually done with a viral genome just to make it easy, small genome that's easily manipulated, and then you then take the viral genome containing a single DNA adduct at a defined site in the viral genome and replicate that in either bacteria or human cells, and then you screen the progeny of those viruses to see what sort of mutations are occurring at that defined site. So the lab that pioneered this work was led by John Essigman at MIT in the US, a real powerhouse of sort of chemistry research, and they were the ones who came up with this very cool strategy for combining the power of organic synthesis and the ability to make a short stretch of DNA that contains a defined DNA adduct at just a single point in that oligonucleotide, a short stretch of DNA, and then they combine that with the power of molecular biology, the ability to generate a genome from a virus, and we call these a gap duplex. And a gap duplex is just made by relatively straightforward engineering, genome engineering, but you just make a plasmid, a viral plasmid that contains an intact circular strand in the inner strand here, and then there's this gap. And that gap corresponds to the length of the oligo, which is designed to be complementary to the bases in the gap on the opposite strand, but it also has this adduct at a defined site. So you can then anneal that oligo against your double helix gap duplex, and then close it up with a ligase, and you now have a nice research tool, a viral genome containing an artificially introduced DNA adduct that you can then go and do biological research on. So you can introduce that virus genome into a cell, look for its ability to cause mutations, and also look at how readily it's repaired and how readily it's processed by DNA polymerase. How readily do they generate mutations? Okay, so this is very cool research capabilities that came out of the Essigman lab, and what it involves then is this production of a recombinant adducted genome containing a single defined adducted base, and very often it's an oxidized base. This is one context where this research proved very useful. Use that to transfect cells, and then you grow out your cells on some sort of plate with an indicator, and often you can use something like a lac-I substrate, very commonly used for assessing the activity of plasmids, modified plasmids in cells. And you can reproduce or use, say, a DNA repair deficient strain, for example, and look at the different yields of mutations in control versus mutated DNA deficient background. And this allows you to do all sorts of experiments. So you can see how is the mutagenicity, the ability of that adduct to generate mutations, how is it affected by the DNA repair background. So you can work out which repair enzymes are patrolling the genome, looking for your particular DNA adduct of interest and snipping it out and repairing the DNA. That's very useful technology. But also one of the really cool things you can do is you can, in human cancers, as you'll probably know from your biochemistry and cell biology, we often find that mutations occur in critical sites in, say, an oncogene, activate a tumour oncogene from an inactive state to an active one, or a particular mutation inactivates a tumour suppressor gene, making cancer development more likely. So what you can do is you can take out the sequence that's known to be vulnerable to mutations in an oncogene or a tumour suppressor gene, and then insert that in the viral genome, and then stick your adduct at a site where you know mutations occur in humans and really matter in terms of tumorigenesis. And so this is really cool, and some research, nice research is being done in that regard that strongly links not just oxidative DNA damage, but a lot of DNA damage caused by tobacco carcinogens. You can do the same sort of technology, make a DNA adduct formed by a tobacco carcinogen and carry it through this sort of technology, and you can show that they can induce the same sort of mutations that are known to be associated with the onset of cancer in a human. Okay, so you can do all sorts of things, and there are multiple polymerases in cells too, and you can work out which one, you can knock out different polymerases in a cell line and then see how that affects the ability to repair or process these lesions and generate viral genomes. Okay, so the sort of thing you can see then, just similar to that other slide I showed you, if you've got an oxidised guanine, you often find that you get a, in this case, a thymine put opposite the adduct, and we call this a C to T transition because we've gone from normally having a C at that site inserted opposite a guanine. Now we've got a thymine inserted there instead, so it's a C to T transition. Okay, so the term transition is used when one pyrimidine is replaced by another, cytosine replaced with another pyrimidine, thymidine, or when one purine is replaced by another. But if a purine is replaced with a pyrimidine or vice versa, we call that a transversion mutation. Okay, so single base substitutions like this can be very important in cancer because they can activate oncogenes or inactivate tumour suppressor genes. Now for the rest of the lecture, I just want to look at some of the actual chemical products of DNA oxidation, and if you're not a chemist you might find this a bit boring, but hopefully will make the point that by understanding the chemistry we get new tools that we can use sometimes as biomarkers to see how much DNA damage is occurring in vivo, or we can do those sort of mutagenesis experiments and work out whether they're likely to be driving the corruption of DNA by generating mutations. So there's all sorts of cool things that we can do once we understand the chemistry. Now one of the major products of DNA oxidation involves oxidative damage to guanine, and this is probably the most commonly used marker of free radical attack on DNA, a product that forms via oxidation of this so-called carbon number 8 in the guanine molecule. Hydroxyl radicals attack this and we get these oxygenation products, which is known, the major product is 8-oxoguanine. But there's also these ring open forms called a FAPI guanine. And you can see what's happened here. is the second 5-membered ring has undergone spontaneous oxidative opening to form this carbonyl species retaining FAPI product. Okay, so this is a very widely used marker of oxidative DNA damage. It's quite mutagenic. It's not highly mutagenic, but it's quite moderately mutagenic. FAPI guanine isn't very mutagenic. The bottom one there, the ring opened one, that's not very mutagenic. It doesn't give any template instruction to the polymerase, but it does act as a replication blocking lesion. So poor old polymerase just gives up when it encounters a FAPI guanine and just leaves that strand of DNA and goes and does something else. So it has consequences for stopping DNA replication, even if it doesn't cause mutations. Now, like I said, 8-oxo-DG, that's the intact sugar with a sugar attached on the base, oxidised base, or the base itself, 8-oxo-guanine, are widely used markers of DNA damage by free radicals. And there's a variety of different methods that people have come up with for measuring this product, and are widely used in medical research. One of the simplest ways to do it is just simply to collect a urine sample, because after this damage gets repaired from the genome, it gets excreted from the cell and eventually from the kidneys in urine, so you can measure the levels of this damaged product in urine. That comes with some complications though, because we eat DNA in our diet, and sometimes we can consume oxidised guanine in our diet, so it just can contaminate your urinary levels a bit, so it's not entirely flawless. But when you account for that sort of basal contamination by dietary sources and just look at how much DNA damage is occurring when you exclude dietary sources, it's still a pretty high level. And there does seem to be an age-related increase in various species. In rats, for example, we get about a 3 to 4-fold increase in urinary 8-oxo-DG adducts with age. It amounts to about resting levels anywhere from 10 to 25,000 per cell in a young animal. So significant amounts of background DNA damage due to basal production of free radicals. Humans, not quite as high as the upper limits in rats, but over 10,000 adducts being excreted in our urine per cell. But there are other analytical problems too to be faced with this sort of technology, allowing for the fact that some of the reagents we widely use in molecular biology to extract DNA and to extract proteins out of DNA samples. You've probably done this if you've done biochemistry. Good old phenol extraction, very commonly used. If you're not careful, if you don't use well distilled phenol, it can cause oxidative damage to the DNA during your workup. So you've got to allow for these sorts of artifacts. Okay, but this is a major product of oxidative damage to DNA, and 8-oxo-DG has a number of different mutagenic consequences that people have identified over the years using some of those site-specific mutagenesis technologies. It can result in formation of AP sites, apurinic sites, abasic sites, but also we can get various types of mutational events occurring as well. I just thought I'd highlight a couple of research papers that I thought were interesting that just showed the sort of things that people do with 8-oxo-DG or the base in terms of using it as a biomarker. This particular study here came from India, and this is some toxicologists used oxo-DG in urine. So they, just to see how much DNA damage is occurring in the work, during the work shift of people working in a fuel filling plant. So people working with petroleum products and other hydrocarbons, there's often contaminants with phenol. Phenols are prone to redox cycling, and this means they can generate free radicals that cause DNA damage, and so there's often been an interest in just knowing how much oxidative damage might occur in a person that's potentially exposed to benzene-related phenols. So phenols form from the oxidative metabolism of benzene in bone marrow and elsewhere. Okay, and these people found that not only did the levels of phenol increase during a typical 8-hour work shift, but also there was an associated, small but in population terms, statistically significant increase in 8-oxo-DG excretion in the urine, and this also is associated with a slight increase in micronuclei frequency. So micronuclei are a form of DNA damage, small fragmented nuclei that associated with oxidative stress. So not a huge effect, but if you're doing this every day of your working life, it's not surprising that some of these occupations have higher rates of leukemia and other types of cancer. Here's a more clinically relevant model paper that people just looked at levels of 8-oxo-DG in human colon tumours. So these are real tumours, and using immunohistochemistry with antibodies against this DNA damage product. And what they did was they compared the levels of 8-oxo-DG in patients that were clinically classified as moderate risk of prognosis. So remember that colon cancer is a very nasty form of cancer, but there are different stages of it, and they can classify people according to their tumour risk and their prognosis. And in a case with very poor prognosis, it literally lit up in terms of 8-oxo-DG in the nuclei of these tumours. So, and this is an indication of oxidative stress making a contribution to what we call genomic instability in tumours. As tumours develop, they become more and more prone to mutations, and they accumulate more and more sequence errors as the tumour progresses. And it's interesting that this occurs against a backdrop of increased oxidative damage to guanine. That's just a couple of papers that just show the diversity of questions, research questions in which the ability to measure 8-oxo-DG has been useful. But guanine's not the only product of 8-oxo-DG isn't the only product of free radical damage on DNA. Adenine also undergoes oxidative modification, although usually it's not as reactive as guanine. But again, you see a variety of products formed. You can see a 2-hydroxy adduct forming over here with the hydroxy group going onto the 6-membered ring in the 2 position. And then also similar to guanine, you get this hydroxylation or oxygenation on carbon 8 from 8-hydroxyadenine. And then there's also this ring open FAPI species. These again are not considered profoundly mutagenic. The 2-hydroxy is probably the most mutagenic, but relatively modest yields of A to G transitions and A to T transversions. And one of the things we see with a lot of these adducts is that the local sequence context affects how much or the frequency of mutational errors. So when the polymerase comes along and hits an adduct, it's not just the presence of the adduct that drives the induction of a mutation, but it's what are the neighbouring bases on either side of the base also affects the mutation frequency in this particular adduct. We see strong neighbouring sequence effects. And this sort of relates to the risk in cancer, where we know certain sequences in tumour genes are particularly prone to mutation, and so Even just moving the same adduct, one or two bases, sometimes can dramatically alter its susceptibility to mutations. And cytosine also, the first of two pyrimidine bases in DNA, also is quite vulnerable to oxidative DNA damage. And again, there's a soup of products formed. I've just identified 3 of them here. There's a 5-hydroxy product form up here. Not particularly mutagenic. But then there's a couple of really nasty adducts formed, and these are called uracils. So uracil is formed when, see that deoxycytosine's got this oxo substituent here, but it's got this amine on the ring, and when that gets oxidatively removed, we get another carbonyl species here. And when there's 2 of them on a cytosine ring, we call that that's called a uracil. And so one of the products of exposing deoxycytosine to free radicals is to generate a 5-hydroxy uracil, and then there's also a uracil glycol, because you'll see that this is a double bond here, and that double bond can be saturated with addition of another water molecule to form uracil glycol. So a glycol is a 1,2 diol, 2 hydroxy groups adjacent to each other. These are very nasty species indeed. Some of the most mutagenic products ever found in these sorts of mutagenicity experiments. They generate mutations in over 80% of progeny, which is really the sign of how nasty these particular lesions are. And then finally, thymine, the remaining pyrimidine base in DNA for us to consider. It also is susceptible to the formation of a glycol. And again, glycol is a 1,2 dihydroxy product. And this isn't very mutagenic. It probably blocks replication, a bit like that FAPI adduct derived from guanine. But the interesting thing is this is a great biomarker because it's chemically stable, and it's been around for a long time. People have been measuring this product for a long time in urine of different animals. And one of the cool things people find is that, particularly if you measure the base without the sugar attached, just a thymine glycol, significant species-related differences. And it seems that some of these small rodent species that can have very high metabolic rates, that consume a lot of oxygen, tend to churn out a lot of this particular DNA damage product in their urine. So this is very old data done by the work of Bruce Ames, one of the pioneers in chemical carcinogenesis, and invented the famous Ames test you might be familiar with from microbiology. But he became very interested in the role of endogenous chemicals and endogenous oxidants in contributing to cancer risk back in about the 90s and 80s, and sort of helped us appreciate that some of these endogenously produced molecules might be causing a lot of the damage that's associated with cancer. Okay, so when we tally things up, bit of an old paper also here, but when people looked at measured different types of oxidation products in the genome of a typical human cell, and I think this was done in a culture cell line, if I remember rightly, but you can see the levels here of most of these adducts sort of in the vicinity of 1,000 to 2,000 lesions being formed per day in a typical human cell. And down the bottom here too, these adducts, these are phenoadducts. I'm not going to talk about these much, but these are formed by adduction of aldehydes formed during lipid peroxidation. I'm going to talk about them in my lecture on Monday next week. So lipids undergo fragmentation by free radicals, a process known as lipid peroxidation. It produces some nasty electrophilic aldehydes which can themselves attack DNA and form some of these quite nasty bases, call them phenoadducts. And so if you add it all up, that's quite a lot of routes to chemical damage in DNA occurring just spontaneously as a consequence of respiration and oxygen utilisation. Now you're probably sitting there thinking, goodness me, I might stop breathing for the rest of my life to save my DNA from being damaged by these reactive oxygen species released from mitochondria. But the good news is we do have the capacity to repair them, and in particular some of them are repaired better than others, and thankfully 8-oxo-guanine is repaired pretty nicely and pretty quickly. It is actually pretty well, most of it's repaired within just 10 to 30 minutes. A half-life for a lot of these adducts is in the vicinity of 10 to 30 minutes. So this just means that within a cell, when these damaged products form, there's a bit of a competition between repair and replication. So the more oxidative damage is occurring, the greater the likelihood that some of them are going to get replicated and introduce a mutation before they're repaired. So that's why there's sort of an age-related increase in mutational consequences of some of this damage. Okay, so this is quite a hot area of research back in the day, and the significance of DNA repair, of course, was recognised in a Nobel Prize for chemistry. Some of these gentlemen had worked out how these sorts of adducts get repaired, and it's quite complicated. It turns out just in guanine alone we have multiple levels of repair, and this just shows us how important it is to biological systems to get rid of this stuff. And it's called the GO system, or the guanine oxidation system, and it involves repair even at the level of what we call nucleotide pool sanitation. So guanine, even floating around as a deoxynucleotide in the cell, can also get attacked by free radicals and then get inserted into DNA by the polymerase. So we've got an important repair capability. MUT is the gene product. It's an ATPase, so a GTPase rather. So it finds an oxidised base and chops it in half before it even gets incorporated into DNA. So that's one level of the DNA's protection against this damage. The one that really matters though is base excision repair. And this is why these adducts only have a half-life of 10 to 30 minutes. This enzyme OG1, or HOG1 in humans, it basically chops out the abnormal base from DNA. There also, if that fails, there's a nucleotide excision repair pathway, and it doesn't just chop out the base, it chops out the 8-oxo-G, the whole sugar and base attached to each other, and a run of deoxynucleotides alongside it on either side. And then if that fails, and if mutational events does occur and the adduct gets replicated and induces a mutation before repair happens, then there's even repair against that. So mismatch repair enzymes come along and see a inappropriately inserted base opposite a lesion, form a lesion site, and say you shouldn't be there, and it repairs that. Okay, but the one that really I think is probably the most important in humans, and we see if you have a mutated variant of this particular HOG1 gene, you do have high rates of cancer and mutations. Just shows how important this is. But basically what happens is after a damaging event by a reactive oxygen species, we get a damaged guanine here, and the glycosylase comes along and chops out that base, and it leaves the sugar phosphate backbone intact. And there are two different repair pathways. One chops out the full sugar and the phosphate group attached, and then there's one that just chops out the sugar, and then a phosphatase chops off the phosphate eventually. So two different repair pathways here, but basically what it does is it makes a cut in the sugar phosphate backbone to correspond to the A basic site, and then this gets filled by a DNA repair by a polymerase and a DNA ligase to close up the sugar phosphate backbone. So we've now restored the DNA synthesis sequence. Okay, so this is probably a very important capability in humans, the ability to repair guanine oxidation, because guanine is probably the most vulnerable base in DNA to free radical attack, and it's very important we have this capability. But what happens during cancer, to return to the theme that I used to introduce this topic at the beginning of the lecture, is that perhaps because of our lifestyles, whether we smoke tobacco, that sort of thing, how much fruit and vegetables we eat, how good our antioxidant intake is, all this increases our vulnerability to some of these endogenous pathways of free radical formation. We get this DNA damage occurring, and it often occurs in clusters. Particular runs of DNA are vulnerable to oxidative damage. If it's not repaired, then we can have persistent DNA damage and accumulation of mutations, and this contributes to this phenomenon of genomic instability and drives cells towards the development of a clinically significant tumour. So that's sort of the basic context for what we're talking about in this lecture then, is that this susceptibility to DNA damage is bad news for organisms, because it means you can corrupt the DNA, or DNA's capability to store information accurately by corrupting the base sequence. Okay, well, thank you very much for your attention. That's DNA, and the next lecture we'll have a look at proteins and how they can also sustain damage. Thanks. Let me just grab my glasses. Yep.